Hello, I am Aiden, and this is my cameraman, Cyrus. Today, we're going to be interviewing the DNA experts, Logan and Chloe, from O'Neill High School. They are going to be walking us through the DNA replication process.
Here we have a simple DNA model.
Can you explain the structure of the model?
Of course. The black and the white structures represent the sugar phosphate backbone. The colored ones, the red, black, blue, and green pieces, are called nucleotides. Nucleotides consist of 3 parts: 5 carbon sugar, a phosphate group, and a nitrogen base. The clear pieces in the center of each nucleotide represent a hydrogen bond. These bonds are responsible for holding 2 base pairs together. The rules for pairing nucleotides together are A pairs with T pairs and G pairs with C pairs.
How does the DNA replication process begin?
The DNA replication process begins with the DNA unwinding and unzipping. Think of a jacket unzipping. During this process, the hydrogen bonds break apart and the nucleotides separate. Pieces of DNA are called RNA. When it gets to this stage, we know it's ready for replication. Once the DNA starts to separate, an RNA primer is added to the parent strand. The leading strand replicates in the 5-3 position.
What is the name of the key enzyme?
The name of this enzyme is DNA polymerase. It attaches to one side of the open DNA strand, which is the leading strand, and works towards the fork, adding new complementary nucleotides. So this would be A and T and G and C.
How does the lagging strand replicate?
On the second side of the open DNA, known as the lagging side, the RNA primer is once again followed by DNA polymerase. Then binds to the open DNA just like what happened to the leading side, except this time we're working away from the fork, creating new nucleotides. During this process, the lagging strand replicates from the 5-3 direction.
And then you'll just have to show video doing DNA stuff.
What is the name of the key enzyme that adds the new DNA nucleotides on the lagging strand?
This enzyme is known as DNA ligase. It's important for sealing gaps in the Ozaki fragments on the lagging strand, completing the new strand. That is how DNA replication works, and this is happening in our bodies at all times.